I'm not actually going to present any epigenomics, transcriptomics, or microfluidics. Instead, as I talk to you about how we have used a very highly immunogenic neoantigen to induce a spontaneous endogenous multi-antigen immune response, you're going to see a lot of mouse survival curves. Towards this way. You might not get to see the survival curves. Oh, there we go. Those of us who do what's commonly known as small data often don't have conflicts of interest. Which direction? Is the battery dead, maybe? Okay, great. So as many of you may know, ALL is actually historically considered a very challenging target for immunotherapy. It doesn't express very many costimulatory molecules. It can have impaired antigen presentation. It can induce anergy in immune cells. And historically, it was tough, but of course, everyone here knows about the tremendous successes that have happened in recent years. Typically, relying you know about the antigen targeting with antibodies, with or without toxin or chemotherapy conjugates, chimeric antigen receptor modified T cells, of course, and bispecific tumor engagers. And what these approaches all share in common is the circumvention of immune tolerance by targeting a single tumor antigen, usually with an antibody. And this has really changed the game. I know this is sort of an overused term for this therapy, but I think many of us in this room have patients who are alive today who would not be alive without these therapies. So these 80 to 90% CR rates are just incredible. But unfortunately, at least with CAR T-cell therapy, as an example, a third do go on to relapse. And you can see here, depicted on the left, is a survival curve of the Penn CAR. Other CARs around the country have similar outcomes. And there are two main mechanisms by which the leukemia does escape from the CAR T cells and other immunotherapies. The first is by development of antigen escape variants, which we heard about earlier today, which is evidence of immune editing by the CAR T cells. The second primary modality is by lack of persistence or loss of CAR T cell expression. It's not surprising to this audience that the immune effect of a CAR T cell is not going to generate immune memory. So in thinking about these problems, we were trying to think of ways to develop a multi-antigen immune response to try and get around immune escape by a single loss of a single antigen. And similarly, by using an endogenous immune response, our hope was to generate immune memory for ALL. The goal for this would be either to use this in conjunction with a CAR T-cell approach, or perhaps instead of a CAR T-cell therapy in cases where there may not be an antigen that is as easily targeted as some of the key ones that have been put into use so far. In thinking about this this goal, we hypothesized that recognition of multiple antigens would be necessary for a durable immune response to ALL. We figured that tolerance to tumor antigens would thwart us, and our hope was that our experience using innate immune stimulants may allow us to overcome some of the challenges that we anticipated. So just to walk you through a little bit our model, so we use the emu-RET fusion protein mouse model developed by Robert Wasserman and Richard Hardy at Penn in the late 90s, I believe. This is a model in which there is a constitutively active RET tyrosine kinase in approximation with the heavy chain promoter. And the mice develop an expanded population of pro-B cells in fetal life that persists after birth, and then they develop additional hits to this population, and this pre-leukemic population eventually presents as a fulminant leukemia between 3 and 9 months of life. We have several cell lines in the lab that we commonly use, and we took one of these lines and transduced it with a dual construct that expresses green fluorescent protein and firefly luciferase, or GFP luciferase. What happens when we put this neoantigen into this cell line, this is our poster mouse, mouse 691, literally a poster mouse. This mouse will, as you can see, these mice develop leukemia that is visible by bioluminescent imaging. This is actually a very low level of disease. This is, if we didn't have bioluminescent imaging, this would probably be maybe 1% or so in the bone marrow, but because of this sensitive method, we're able to detect it. And then the mice go into a spontaneous immune-mediated remission by day 10, and this is durable. Survival after receipt of this type of leukemia is typically between 75 and 100%. There are some rare late antigen-negative relapses and some earlier antigen-positive relapses that I'm happy to talk to you about after. And just for reference, this orange curve that goes straight down represents the wild-type leukemia. It kills you by day 35. And the green curve is the GFP luciferase leukemia. We explored this a little bit and discovered that this is, in fact, a T-cell dependent immune response. So T-cell deficient mice, whether they be antibody depleted or RAG knockouts with or without an additional gamma common knockout, these mice are not able to control disease and all die early on. This is an example of their development of luciferase positive disease, and you know the straight line down. Natural killer cell depletion early on does not actually lead to disease breakthrough. Interestingly, we see this breakthrough with either CD4 or CD8 depletion, suggesting that there may be some cooperation between the two. This is a transplantable T-cell response. So if we take a mouse who has, a BALB/c mouse who has cleared disease, remove the spleen, sort the T-cells, and transplant them into a RAG knockout, we do get complete protection from the GFP luciferase. If we take splenocytes or T-cells from a naive mouse, we get a partial depletion. This is probably just because we put in the whole bulk population of T-cells. We didn't select for epitope-specific T-cells. And then, as you can see, the RAG mice who don't get T-cells all die of disease early. Tolerance to the neoantigens will abrogate this response. So we have BALB/c transgenic mice who bear GFP luciferase in every cell. These mice are unable to reject disease, the red line, they're unable to reject disease as compared to our wild-type BALB/cs depicted in blue. Interestingly, when we put the luciferase-bearing leukemia into our emu-RET transgenic mice, what you can see here is the red line down is the wild-type leukemia going into both wild-type BALB/cs in the solid red, and dotted, the red is the emu-RET transgenic mouse. They all die around the same time, so there's really no difference in survival. If you put the GFP Luc-bearing leukemia in, you get complete clearance in the wild-type BALBs, and you get a delay of disease onset in the emu-RET mice. And this simply suggests that the immunogenicity of the GFP luciferase does allow for some recognition by the immune cells of the emu-RET mice, but the emu-RET mice are likely tolerized to an emu-RET-associated antigen on the leukemia cells, and they're unable to completely control the leukemia. We can partially rescue these tolerized mice with adoptive transfer. So if we take a BALB and have a clear leukemia versus naive mice, we can T-cell sort, put them into our GFP Luc transgenics, and then re-challenge with GFP-bearing leukemia cells. you can see that the mice that get naive T cells in yellow look just like mice who got no T cells. They all die quickly, whereas mice who received educated T cells have a bit of a prolongation in their survival. And this is not definitive evidence, but is potentially suggestive that our initial hypothesis, which was that these heavily tolerized mice would have complete central tolerance, just putting back in T cells with the potential capability to recognize the antigens is not a complete rescue, and so there may be some additional issues with peripheral tolerance in these mice. And we recently received some funding to explore this a bit more. What we didn't expect to see was that the neoantigen response actually appears to induce some epitope spreading. So if we take our mice who've cleared, who've cleared their leukemia, biopsies who've cleared the GFP luciferase-bearing leukemia, and then re-challenge with wild-type ALL, these mice almost always clear this leukemia and reject it as well, which was an unexpected finding for us. And we attribute this to presumably a multi-antigen immune response. There's no GFP or luciferase to recognize in these cells, so they're clearly recognizing something else on the tumor. This effect seems to occur early, so if we, irrespective of whether we clear the leukemia and then re-challenge, or if we give a one-to-one mix up front, the mice are able to recognize the additional antigens and clear the disease. Scott is thinking about another non-antigen-targeted, durable, adaptive immune response that we had seen previously. We had given, several years ago, we used a TLR9 agonist, so innate immune stimulation, to induce an immune response against syngeneic non-GFP luciferase-bearing ALL, and we found that not only did mice who were treated with CPG clear an initial adaptive transfer of leukemia, they were also able to reject a re-challenge. And you can see here that naive mice all die of leukemia, whereas mice who had previously experienced CPG had an adaptive immune response. So we were hopeful that we might be able to overcome some of the tolerance issues we had by doing innate immune stimulation. This is a little bit of a complicated figure. So what we did was we took 3 transgenic strains. We have our GFP Luc transgenic strain, RFP, which is our lab's shorthand for the emu-ret fusion protein transgenic strain, or dual transgenics. And we gave them GFP-bearing ALL. We then treated them with CPG or not. And you can see that I'm going to actually start with the GFP transgenics in yellow in the middle. Mice were well on their way to developing their disease in the PBS arm. When they were treated with CPG, there was a significant difference in disease burden. There was a significant difference in all 3 arms, the difference being that mice who were dual transgenics had a much lower decrease in disease burden. The significance of the emu-ret transgenics is less marked just because they hadn't developed as much disease, because likely that GFP luciferase is stimulating enough that the PBS arm were controlling disease on their own without the CPG. Unfortunately, while this did translate into a survival benefit, it did not completely overcome tolerance to the neoantigens. So hopefully, for now, I hope to have convinced you that we do feel that a multi-antigen response is required for endogenous durable immune control of leukemia, and that antigen-specific tolerance will undermine immune control. We did observe that innate immune stimulation reduces disease burden but doesn't overcome tolerance. We hope that our tolerance models overshoot the tolerance bar in children, but certainly more work needs to be done. Obviously, I think we'd really be interested in identifying how we're inducing epitope spreading with this neoantigen. The neoantigen itself is not going to be translatable, but if we can figure out how we're getting this immune response, hopefully we can co-opt that for kids. And as I mentioned earlier, we are exploring some approaches to circumventing peripheral immune tolerance by looking at PD-1 and CTLA-4 blockade, Treg depletion, and CD40 ligation. And with that, I will thank my outstanding lab tech, Jessica Lee, my colleague and collaborator, Gregor Reed, and his student, Suman Jo, have been incredibly helpful with this work, and my mentor, Steve Grupp, and colleague, David Barrett, everyone in the Grupp lab, and my funders. Thank you.